At the outset, I would like to thank the Secretary DBT, Dr. Rajesh Kokolay, for making time. I know he's very busy with stuff, but he still took out time to address the symposium. And before I request him to make an address, I will just share the slide about NII, because NII, I mean, of course, most of you are no strangers to NII, but there may be some people who do not know much. Just one slide, couple of minutes of introduction, and then we move on to the business of the day. So if we can, just share my screen. So I hope it is visible, right? Is it visible? Is it visible?
Yes.
No, it's still not full screen. Huh? It's not full screen. Yeah. So this is where we are. I mean, in the heart of New Delhi, but still probably the greenest part of Delhi. National Institute of Immunology moved to this building almost 3 decades back, but it was initiated or started from All Indian Institute of Medical Sciences by Professor G.P. Talwar. And one of the first reasons why it came into existence probably was the need for anti-leprosy vaccine in the country, and so on and so forth. And it is an autonomous institute of Department of Biotechnology, and the oldest institute of Department of Biotechnology. It actually predates DBT. And from the initial forays into vaccinology and reproductive immunology, NII grew and diversified its research program to understanding aspects of basic immunology, infectious diseases, started working on pathogens ranging from TB to malaria, leishmania, and so on and so forth. And recent decade and a half, we expanded our research areas into understanding other areas of human biology and disease, such as cancer, which is a major activity now at NII. And of course, now is the time where we're trying to get more into translation and public health, and going forward with that. And of course, continuing to do what we have been doing. We want to build programs which are centered around some of the areas I mentioned, and just to show that that currently we're involved in some of these areas, which are indicated on the slide here. It would have been great if we had all met in person, but nevertheless, it's a good occasion to meet by virtue of the symposium, and I look forward to very interesting 3 days. Just a brief pointers about the organizer. Shauman Basak, Veena Patil, and Rahul Pal have been pivotal from NII's side to put together this program and make arrangements. But one of the major reasons this symposium is happening, and in this format with the array of speakers of high quality and experts in the area, is Professor Shiv Pillai. My only contribution probably to this was when I sent an email requesting Professor Pillai if he could speak on a symposium, and also if, given his connections and stature in the field, if he can help us put together a program. Within half an hour or so, I got an approval. Yes, I would like to do. And then a few minutes later, I got another mail. Oh, Pushkar, I noted your second request. I'll be able to help you out with speakers and so on. And that triggered it all. And within a short span of 6 weeks, we were able to put this program. Thank you very much, Professor Pillai, for that. I would now like to welcome Dr. Rajesh Kokolay, who has been our colleague at NII for almost 2 decades. Most part of this last 2 decades he has spent at NII as a scientist. His contributions, especially in the area of understanding pathogenesis of TB, are something we're very proud of. Just this morning, his discovery of these QPR4s or zinc binding molecules from MTB was published in PNAS, and was very excited and congratulated him, and so on and so forth. And Rajesh has been, that is only one aspect of his research, but he has been working on vitiligo and several other important areas of biology. And I would not have the time to go into detail, and he would not like knowing him that he just wants to get with the business. I mean, I will not elaborate much, except I will say it was also a great pride for NII when he took over the charge of the Secretary of Department of Biotechnology in September. And we can already feel his energy and the vibration which come into the system, and he continues to now motivate us, which he was doing even as a colleague, but now in a different capacity, and we look forward to his support and leadership. So with those words, I will request Rajesh to say something to us, and I'm sure he will have something important to tell. Thank you, Rajesh. Over to you.
Thank you, Pushkar. And I think it's thank you all my NI dear friends, and Professor Pillai. Thank you very much for organizing this. I think it's such an important time to continue to carry out these dialogues, and all the students and community from various places who have logged on to it here. Let me say thank you very much for inviting me here for this webinar series. I think this has been an extraordinary time and an extraordinary level of activities that have been catalyzed across the world that really has brought what I should say scientists and governments and industry closer than ever before, actually. And this has been the most satisfying aspect of, if you really ask what has been the most critical aspect that has worked really, really well actually in this period of time is despite differences that we always had across these three very important domains of systems, we've been able to come together and deliver in a way that is actually quite outstanding. We've not only discovered vaccines, we've not only delivered vaccines, but we deployed them to such a large level in this country, which is super and completely outstanding from a variety of perspective. Of course, if just once looks at back, 2 years back, and try and think of the uncertainty with which the whole things had started. I mean, there were, as you would all say, that there were clearly, to begin with, there was a denial at the part of government as well, and many of the scientists that this can indeed be true, because there was as you must have all of you seen a movie actually before, which on pandemic that had the Hollywood movie that had come, Contagion, and actually it almost reverberated what happened in that movie. Actually, you can see that. So it's very, very interesting to look at, and I think clearly it's been even though we think there are lots of good things that have happened, but there are clearly been a most challenging period of history of humankind, at least going to be in our times. Actually, more than 5 million people died all across the world, and this is almost 1/10 of what happened in Second World War. Actually, so it's unprecedented to think of it, of what magnitude of things have happened. It's not only this, the lockdowns and all variety aspect, which is the sovereignty of human life, was in fact taken away. If you really look at from a point of view that you are restricted to move around here. So it's been a very, very trying time in human life, and with uncertainty that what we should do, how we should proceed, starting from, where this virus is going to pass across and how we do it, and to a position that we today are, and hopefully, we hope we have some control over the situation right now. And I think Professor Shiv Pillai can tell us much better than what I can, because I think he's in the midst of variety of these things in different ways. But yeah, I think it's just amazing that how it has catalyzed variety of important activities able to do. And what I really think is that now that having been done very well and broken some of the barriers of really rapid development, what we've been seeing, which always takes years to come by, I think it's been a really, really fascinating aspect of how we can continue to do these kind of activities in future to come. And in fact, a friend of mine was mentioning it to me. It looks like, just saying, maybe all the, obviously the scientific commitment that has come, scientific thinking that has come. They said maybe all the clinical research regulatory committees should become like venture capital mindset. Actually. And he was telling me that's probably the only way one can look at it. It's very interesting perspective, because as venture capitalists look at various, when they try and evaluate a thing, it's always based on probability of something to happen, actually, right? I mean, the financial predisposition is all really pulled out, actually. So can the regulatory in biological space, in fact, function like this, and can therefore, really enhance the pace of what one wants to look at, and can, in fact, this leads to kind of dramatic changes in the timelines of what biological products that reach to the market. I mean, it's something that we need to discuss about and think about, but it's kind of an interesting thought that somebody, a friend of mine, suggested in one of the meetings. I think I would just say, just stop here and say clearly, this is such a relevant topic, and it's wonderful, Pushkar, you organized this COVID symposium. Better late than never, actually. Maybe this is the perfect time because we can say that maybe we probably are looking at the fag end of which I've been saying, this pandemic. Of course, there are new variants will come, but even if the new variants will come, we now are better prepared to handle. We have better understanding of what we should do and how we should go about doing it. And you may find different clinical manifestations, but I think these are, again, not as dramatically unknown to us as what have been there actually. So I think I will stop here, and I think you have galaxy of people who are very well, far more experienced and learned and far more articulate and have come with their own wonderful things. So really, thank you again. And I wish I can attend the whole of this meeting, but I'll be here for at least Professor Shiv Pilla's your talk, and hopefully I can sit through this and enjoy your wonderful lectures that you give over the years in India. I've heard before that as well, actually once on some years back. And I think hopefully it'll be a wonderful session. Please keep me updated if there are some interesting things that come out of it. And I think I'm pretty much sure students and other faculty members and colleagues from all the place who have joined actually is going to have a wonderful time discussing over the next 3 days. So thank you very much again, and I'm delighted. I hope I can be at NII. I mean, this is nothing better than listening to wonderful science in life, actually. That's the most amazing thing of what I miss really is that part of life. Hopefully there are other contributions that I can also make from this directions. And just to say a brief word about Department of Biotechnology, actually, because now that I wear this hat, I need to say a little bit good things about DBT, actually, what they have been able to do, actually. So DBT have extensively supported startups to do large number of diagnostics that at the initial phase was very, very critical, actually. If you need about more than millions and millions of diagnostic kits, you need to build capacity in the country to have large number of people to be able to do. So more than about 100 startups were supported to make diagnostic kits very, very early on, which actually catalyzed these activities. Of course, what DBT has also done is supported several vaccines, including DNA vaccine, including the mRNA vaccine, including the vaccine, the Covaxin augmentation programs and variety of other things that have been very, very critical and handhold and support, actually. Hopefully, many of them actually will also come to the market. Recently, the Corbevax, the protein-mediated vaccine, also has been given an emergency approval. And finally, the genome variant tracking actually has been very, very interesting experience for us to understand that how viruses, in fact, go through this pandemic processes and how over the 2 years these processes occurred. So I think it's exciting to be at Department of Biotechnology right now, and I think I'm probably a little bit starting to feel as part of DBT, although I miss NII a lot. But yeah, it's an interesting experience. And again, I hope I will try to attend as many lectures as possible on my cell phones, actually, and hopefully I learn a lot more out of it and become more and more interesting for me. So thank you very much, Pushkar. I think it's really great pleasure, and thank you for inviting me.
Thank you.
My best wishes to all of you. Thank you. Bye.
Thanks, Rajesh. Thanks a lot. We're getting time, and I would just say it is a great opportunity, even though the whole COVID story for pandemic has been of great human tragedy, but it also is a reflection of great advances science has made, be it vaccination or the valor of the health workers and so on and so forth. But what it's done is it has ignited young minds and kind of questions, the kind of interests, especially in immunology, which we find from young students, which we earlier didn't. I mean, it's tremendous. It's exponentially increased. This is exactly why, if a symposium like this can ignite some minds, we would have done something more than usual, which is to do good science, which obviously is most important. I will request Shaman Basak to take over the proceedings and introduce our first speaker. And thanks very much, Shaman, please.
So a very warm welcome to all. Well, COVID-19 pandemic had its own detrimental effect on our livelihood for sure. It also provided an elaborate opportunity to revise and advance certain basic tenets of immunology, particularly those that are associated with infectious etiology. And we are fortunate that here we start with no other than Professor Shiv Pillai to learn about those recent advances. Professor Pillai studied medicine at CMC Vellore, received his PhD degree from Calcutta University under the supervision of Professor Bimal Bachet, and was a postdoctoral fellow with Professor Dr. David Baltimore at the Whitehead Institute and MIT. He's currently a professor of medicine and health sciences and technology at Harvard Medical School. He's also the program director of an NIH-funded Basic Autoimmunity Center of Excellence at Massachusetts General Hospital. And he is the director of Harvard Immunology PhD program and Masters in Medical Sciences program. Professor Pillai actually indeed, as Pushkar was mentioning, has received numerous awards, including those related to teaching and mentoring. And he interestingly originally coined the term surrogate light chains for describing proteins that he identified as a part of pre-B receptor driving early B cell development. And his subsequent work indeed indicated the role of BTK in ligand-independent signaling models. And his current work on COVID-19 suggested that a block in T follicular helper cell development in COVID-19 may prevent the formation of an effective germinal centers. Today, he will discuss critical alterations in the adaptive arm of the immunity in the context of SARS-CoV-2 infection and vaccination. With this, we request Professor Pillai to take over. And sir, please, you have to start your presentation and unmute yourself.
So I'm sharing my screen. Can you hear me?
Yes, sir, we can.
Okay. So just let me. All right. So I'd like to thank Pushkar and Shomin and Veena and all the organizers. I'm sorry if I haven't named everybody. And I'm also thankful to my colleagues who agreed on pretty short notice to come and speak, especially those, including Dwayne, who I see is on, who have small kids and are on vacation week this week, but are joining from somewhere else. My connection to NII goes back to the 1980s, I think from the days of its inception when I was a PhD student, and I've visited the NII many, many times. And so I have fond memories of the place, and I'm so glad to see that the NII is now at the highest levels of the DBT with its connections, and this is great for immunology overall. So as was mentioned by Rajesh, that this has been a strange time. It's been a time when we have, the world has suffered. We've all had a difficult period of time, but we've also learned a lot from disease and from thinking about it. I'm going to tell you a little bit about what we think we have learned about the immune system. And one of the goals, of course, is to study the disease and understand more about human immunology in general. So I'll start by thanking the people who are involved, and I'll come back to them at the end. But I just want to remind everybody that when we think about T-dependent B-cell help, we have an initial phase where we have what we call an extrafollicular focus. And this is where most isotype switching occurs other than for IgE. And subsequently, we have the formation of a germinal center. So there's the extrafollicular focus and this germinal center reaction. And all of you, I think, are aware that the germinal center is where there's this Darwinian process of somatic hypermutation, selection of cells. Earlier on in the germinal center, we have the egress of memory cells. So they are not very high affinity. They have a moderately high affinity, and they tend to also have more breadth. And we'll come back to the issue of immunological breadth. So one of the purposes of somatic hypermutation is to give you high affinity, long-lived plasma cells, but it is also to create some breadth in your memory repertoire so that maybe we can handle slightly different pathogens. So in the early days, as Shomin pointed out, in the early days of the COVID pandemic, we found that there was a loss of germinal centers. This is to show you that if you looked at thoracic lymph nodes from somebody who did not have COVID, you would see beautiful germinal centers here in gold. But in COVID, you lost these germinal centers. And this is quite dramatic. But one of the interesting findings we noticed at that time is that when we looked for RBD-specific B cells, and we're looking in people who don't have germinal centers, and pretty much we can establish that, and we could see both in the lungs and the lymph nodes and in the blood, we could see B cells and which bound RBD, and many of which had a phenotype which was CD27 positive, IgD negative. So most of them were IgGs or IgAs. And it became clear that there were a lot of these memory cells that we always called memory cells that are formed in the absence of germinal centers. Now, this was not a total surprise because in the mouse, if you knock out BCL6, you can still get switch cells, and we just call them switched memory cells. So we started to wonder whether this is the same type of memory cell that is produced when you have a germinal center, or is this a different type of memory cell? So one of the thoughts in our minds was maybe, when you vaccinate, you have a proper germinal center formation. This has been validated by many people. And then you get switched memory cells. And maybe these switched memory cells coming out of the germinal center might not be identical to the switched memory cells that we have been looking at, which are IgG CD27 positive or IgA CD27 positive, but which we knew could come out of an extrafollicular response. So this was the question that we posed ourselves. One of the questions was, do we have two types of memory, or is there one type of memory? Is there a difference in their quality? How do you define memory? And in human studies, everything is called memory. Everything is called memory, atypical memory, and it's not always been established whether the cells we're looking at are truly persistent cells. Okay, so now there were a couple of interesting studies. So Duane is going to speak after me. So Duane did show that some people, and Michelle Nussenzweig showed the same thing, that as you go into convalescence, some people will develop germinal centers. So you have, he called them decayers and sustainers. So early in COVID, in severe COVID in particular, you don't have germinal centers, it's extrafollicular, but you can later on get germinal centers if you recover. And now we can understand there's viral persistence, and that might be why we still get germinal centers later on in some people. Now, there's another interesting paper that has come out from someone else who was once our student here, Scott Boyd at Stanford, and this is just out a couple of weeks ago. And what Scott has basically shown is that he's confirmed that, okay, in severe COVID, you don't have germinal centers. After vaccination, you have germinal centers. This has been shown by others as well. But he showed that there was broader variant recognition with vaccination compared to COVID. And he compared different vaccines, a whole bunch of different vaccines. And all vaccines had a broader immune repertoire, an antibody repertoire, than COVID itself. So this is an interesting correlate as well. So the question we wanted to ask, are there two durable, different, functionally distinct types of memory? Maybe one is extrafollicular and the other is from germinal centers. And can we use some unbiased approach to look at memory and understand this process a little bit? So what we did, one of the approaches we used was to take vaccinated people, use basically staining with tetramers specific for spike. So these are different flows, use multicolor flow, looked at populations. And when you look at populations, we can see multiple populations. We can break them up a little more. So there's like 14 different B cell populations we're looking at. And these, you can see the difference between pre-vax, which is in light blue, and post-vaccination, which is in purple. Okay? Now, what criteria did we use? We made some criteria, and one criteria was that if something, and we're calling them precursors now rather than effectors, but if something was a precursor, it was transient, it was transient, lasted for a couple of months maybe. The phenotype transcriptionally would be of lymphocyte activation. There wouldn't be an anti-apoptotic signature. Whereas if it was a memory cell, it should be persistent. And I'm going to say this is up to 1 year now. I'm showing you data today from 7 months and a little later, but we've looked up to 1 year now. It should be persistent to call it a memory cell. It should be a resting phenotype of cell. It should have an anti-apoptotic signature. And ideally, it should dynamically change when you boost. So if you're a memory cell, you're supposed to be activated and give rise to plasma cells. So then there should be a dramatic dynamic decline. So these are the criteria, and I'll go through all this as we go through the story. Now, basically, transcriptionally, we're saying that if you are a precursor, you'd have an activation phenotype. If you're a memory cell, you'll have an anti-apoptotic phenotype. And I'm just showing you a little bit of transcriptome data from other stuff that we've done. So I'll walk you through this. So when we looked at different populations, we found that there was a population of cells. I'm only telling you about a couple of markers. There are multiple markers. I'm going to give this a name in a little bit. We had cells which were CD27 low. And if you follow through here, so this is vaccination, multiple doses. And you can see that at the time of the third dose, which was actually 7 months, so it's 6 months after this second dose, 7 months of the beginning, you can see a lot of cells have accumulated of this phenotype. Okay, so we are saying, okay, this is persistent. When we give a boost at this time point, 10 days later, these cells have declined in number. So it kind of fits like being a memory. So it's increased in numbers and has declined. We looked in acute COVID, not too much. By convalescence, we see more of these cells. So it's not really seen in acute COVID. But when you vaccinate convalescent people, then you see this is the convalescent COVID patients who are vaccinated. This is the time they got a single dose. These numbers went down. So it seems like a dynamic population, which is persistent at least for 7 months. And I'll contrast this to others. Now, when you look at the classical memory cells, now we are again using more markers to break CD27 positive cells up. So what we call resting memory in the CD27 pool are cells which gradually increase. So this is vaccination. There's some decline every time you boost. There's a decline, as I showed, for the other 27 low memory population as well. And then when we look now at the 7-month time point, they have increased, not dramatically. There's a slow increase and sort of tapering off, but with the boost, there's a slight decrease. This is what we can see. Now, similarly, but if you look at COVID, in convalescent COVID, these cells are quite prominent. Okay, so what is the most prominent memory cell in convalescent COVID? It is these guys. It is the CD27 positive resting memory cells. And there's a slight decline after boosting. Okay? Now, I'm now showing you what a population I'm going to call a precursor. This is the precursor of most interest to us. So this is a CD27 positive, 21 low. Again, I'll give you slightly different names as we go on. But this cell has been studied before by Patrick Wilson. And he's looked at it in a flu context. But what we see here is these cells, you can see that every time we boosted, they increased and they went down. So if we looked at about 2 months, so this is 8 weeks after second dose, so this is about 3 months, they're gone. They're transient. They don't stay very long. But then, and now you look at 7 months, they're not there. There's hardly any of them at all. But you boost, they go up. Okay, so we look at this as a population of precursors of plasmablasts that emerge upon boosting. Okay, and I'll try to make the connection between the CD27 low memory population and this precursor population as we go through. Okay. The other thing I'm just going to touch on this, I won't spend time on this. We have looked at many other potential precursors. These have all been called memory in the literature, but all of these cell types, these are all 27 negative, are transient. They come up, they go away, they're gone. Okay, if you just go and look at the 3 or 4 month and the 7-month point, they're all gone. Okay, so all of these other populations are transient. This DN3 is kind of interesting. I have a whole story on it. I won't tell you that today. And these tend to be more COVID specific than anything else. So these are cells, we call them DN3s. They're more prominent in diseases. Okay, so then our reasoning was, so it's known that the S2 part of spike cross-reacts with common cold coronaviruses. RBD of spike does not cross-react with common cold coronaviruses. So the reasoning was, okay, if there's a memory population, then maybe there were already some cells around before people got infected or vaccinated, and maybe those cells can recognize the S2 part of spike, but they should not recognize RBD. So sure enough, now we are looking at people who are unvaccinated, who don't have antibodies, they're never infected, not vaccinated. And if you looked at spike-specific cells, you can see they are using 2 probes. So you can see the spike-specific cells showing up. So there are spike-specific cells, and these cells, you look at them, they are in those 2 memory populations, largely speaking. Okay, so there are these cells, they exist, they're against spike. Look at the same people for RBD, there are hardly any cells that bind RBD at time 0. So the RBD immune response is going to be created by vaccination or disease. Before infection or vaccination, there are really hardly any B cells that bind RBD. There are a few, they're naive cells. That's how the response starts, but there are very few. Okay, so this fits with saying, okay, those are memory population. We call those 2 memory cells, and yes, they're there against probably S2 of spike, and they pre-exist. And this is something we've extended further, I'll tell you about later. Okay, so here's now, we said, okay, fine, we're looking in different people. Let's look in the same person, and let's look at the issue of dynamic decline. So first, let me point out, this is pre-boost over here and post-boost. And we're looking at total RBD binding B cells as a fraction of all B cells. It's hardly changed after boosting. There's no big increase. 10 days after boosting, there's no big increase. And this can be explained by the fact that the CD27 low memory cells in each individual, so we're taking before and after boost, declines when you boost. The CD27 positive memory cell doesn't show dynamic changes. The precursor that increases most is the CD27 positive precursor B cell. So this is a transient population. It increases after boosting. So it's reasonable to think that, oh yeah, these cells decline, those cells increase, these are memory, these are precursors of plasmablasts, and maybe they all add up to why we see these changes of not much changes in RBD binding. So the big picture view, and I'm going to sort of tell you some details that I'll get into in a minute. We think there are two types of memory cells. We think one comes from a germinal center, and I'm going to call them CD27 low exist high switch memory B cells. And these memory B cells, if they were activated by the antigen, they would give precursors, which we will call GC type plasmablast precursors. And I should tell you that the cells that I described as the precursors that seem to come out of these memory cells, these cells have been described before by Patrick Wilson, and he has also suggested, because they're highly mutated, that they are precursors of plasmablasts coming from a germinal center. And we presume these cells will give us long-lived plasma cells. The resting switched memory cells, which are 27 positive, will give us some precursors, which we are calling extrafollicular type. This is a biased view because it's all based on what I'm going to think about and show you data for, but it's not absolute. And these will give you short-lived plasma cells. This is the thinking. This is the way we're going to go about, and I'll explain why I'm calling these cells CD27 low, exist high in a bit. So we did another approach. We used single-cell RNA-seq. We took people before and after vaccination, and we took patients with COVID. We used 10x, we used site-seq, we used BCR-seq. We had good collaborations with a core at AbbVie, and we looked at transcriptome, somatic hypermutation, connectivity plots, trajectory analyses, and so on. So just to walk you through this, here is a baseline vaccinated pool of all the people at baseline, pool of all the people after vaccination. The first thing I want to tell you is that this pool here of 0, 1, and 2 that I'm showing here are your 27 positive resting memory. This pool of smaller pool here of called number 5 is your CD27 low resting memory. Then I'll tell you why in a minute. After vaccination, we see a new population here called 4, which are those precursors that I told you about, which are 27 positive, 21 low. Okay. These are number 6 here is the extrafollicular precursors, which have increased. Number 7 are cells I'm not going to get into today, but in the literature, we call them activated naive. They come from the naive population, which is number 3. Okay, so I just want to lay this out for you and say there are these populations. We have looked at the surface markers and tried to correlate them. Then we looked at transcriptome as well. So again, the big picture view is, I'm saying there's a canonical 21 positive memory, and there's a resting, this is also resting, and a resting CD27 low, I'm calling it exist high memory. These two memory populations pre-exist, you activate, you get this pool, which I'm suggesting comes from number 5, and this pool, which I'm suggesting comes out of 27 positive, CD21 positive memory. Okay. Now, when you looked at transcriptomic data, and this told us a lot, so the memory population that we call CD27 low switch memory, one of the striking markers is a long non-coding RNA, well understood in terms of X inactivation, called exist. In fact, it's the first long non-coding RNA ever discovered, and I'll talk a little bit about it in a minute. And there's another transcription factor called KLF6. So this is how we are defining this population. The resting switched memory in these pools is more directed towards what we would call a plasmablast-like phenotype. And these are the activated cells, number 4, which have an activation phenotype. All of these have lost exist. And then we also have an activated cell type that is more extrafollicular in population number 6. We use a trajectory tool. There's a limit to saying how much this tells us, but I think it's reasonable. And it fits in with saying that this is the pool of resting CD27 positive switched memory. It gets into the extrafollicular plasmablast-like pool. Again, take my, I have to say that I'm basing this on transcriptome. And then this is the 27 low switched memory, which is giving rise to those precursors, which I said are precursors of plasmablasts likely of germinal center origin. So this is the way we see it. It fits in that extrafollicular will be before germinal center, and this will be the trajectory transcriptomically. You can actually see it by looking at the transcriptome a little bit, but this is the way we are thinking about it. Now, when we looked at somatic hypermutation, we're looking early now, right? Soon after vaccination, early acute COVID. There are two clusters which are highly mutated compared to everything else, meaning more than half the cells. So less than half the cells are unmutated. The blue is unmutated. And one is cluster 5, which is that resting CD27 low exist high memory population. This is all within 5 weeks of the first dose. Okay? And this is the CD27 positive to 21 low precursors. Again, same time point. It's about 10 days after the second dose, about 5 weeks, 6 weeks after the first dose. So these two seem to be the most mutated. So we consider this to be fitting with the germinal center origin. Now there's better data on flu because, it's been looked at in more depth for this population. This is what Patrick Wilson also suggests, post-germinal center cells. Okay, so there's a paper that came out last year from Howard Chang at Stanford. And Howard showed, and this is really cell line data, but it's very interesting. He showed that exist, which is important for enforcing X inactivation during development in women, is also relevant in somatic B cells. And the relevance is for certain X-linked genes which are kept suppressed. And he suggested TLR7 would be a good candidate because he looked at it, at least in a cell line data. And he showed that how you could have X inactivation of TLR7. He didn't really define the cell types well because he's looking at a cell line. So we now put this together and say, this kind of makes sense. If you lose exist, so if you have exist, you'll be in the resting phase. We'll be suppressing the expression of TLR7 among the other things. If you lose exist, you're now going to have the activation stage. And now is when you get those plasmablast precursors. That's the way we think about it right now. And there are other pieces of data which would support this. So just to summarize, I've told you about two types of memory cell. There's a CD27 positive memory subset and CD27 low memory subset. This has a different signature. There's a higher level of mutation. It's prominent in vaccination. It's more dynamic. And does boosting increase memory? Yes, indeed. This is when we looked at boost and we're looking with RBD, we saw that most of the cells that bound wild-type RBD, at least 60% of them also bind Omicron. So all Omicron binding cells will bind wild-type, but these don't really increase much with boosting. But when we look at whole spike, when we're looking at spike now, you can see that, yes, the cells that are bind wild-type bind Omicron, and there's an increase after boosting. More interesting, if we actually looked at these cells in somewhat more detail, so this is before boosting. You can see we've divided it by MFI, so brighter versus less bright. Before the boost, there are fewer cells which are highly bright. After boosting, there are more bright cells, and we show that these cells are the memory population, and these cells are the precursor population. This is what we are calling the CD27 low exist high memory, and these cells are largely the CD27 high 21 low precursor population. Yeah. And this increases after boosting. You see an increase in cells that bind Omicron. So this makes sense overall from the literature that there's a memory population that's giving us diversity. There are these two memory populations. We think they are of different origin. Obviously, our data doesn't address that directly. Okay, so there are a couple of related questions. I'll go through them quickly. One question is, I'll skip this first question. Is there a host protection related reason for loss of germinal center? What is the mechanism of TFH loss in severe infection? And so this brings us to what's going on with cellular immunity. I'll go through this quite quickly. We've already published there's a loss of BCL6 positive TFH cells. But why does this happen? What is the reason for this? Why does this happen in severe infections? Mechanistically, we are studying this in the mouse, but we did show that there's a loss of TFH cells. It was pretty dramatic that you lost BCL6 positive cells. You could look at this by quantitation per square millimeter or by proportion, and there was a loss. Okay. Now, we also posed, this is known from mouse data, that if you had a lot of TNF alpha, you would probably lose TFH cells in germinal centers, and it kind of fits that idea. But then why do we do this? Why is it in severe infections we actually lose TFH cells? And so the thinking is that what we think happens is that with severe infection, you divert your cells from becoming TFH cells towards becoming CD4-CTLs. And in severe infections, there's a reason to actually dampen CD8 activation because you don't want to lose an organ, but you depend then on cytotoxic CD4 T cells. And what we show and others have shown, so Veena will be aware of this work from her old lab where the CD4-CTLs have been seen in the blood. We found CD4-CTLs also in the lung. They're the most prominent CD4 cell in the lung in severe COVID. So I'll walk you through this quite quickly since time is short. So yes, we see that there are Grand-Zambi CD4-CTLs and CD8-CTLs are in equal numbers. CD8-CTLs are not increased in massive amounts as you would think they would. It's actually incredibly low. And the large proportion of CD8 cells express PD-1. So if you looked at PD-1 expression here, we're looking at PD-1 and CD8 cells. This doesn't prove that they're exhausted, just tells you that phenotypically they might be exhausted. There aren't many of them. They don't all express, very few express Grand-Zambi, and many of them express PD-1. But there's lots of CD4-CTL. Cytotoxic CD4 cells are increased in COVID with time, and they are the most prominent CD4 T cell we can quantitate in the lungs. And they also increase in the blood with disease severity. This replicates what has been seen by other means before. They're known to be antigen specific. There's a lot of class 2 expression in epithelium and endothelium in the lungs with severe infection, which will explain how a CD4-CTL might work because it needs to see class 2. There's increased apoptosis. So you can see when you're looking at cleaved caspase 3, you can see quite a lot of apoptosis in the lungs. And so the big picture view we think is that, we don't need TFH cells in a time of very severe viral infection. We need to clear the virus. So we give up on making TFH cells, divert cells. It's a sort of immune deviation to go as CD4-CTLs. And this probably helps us to clear infection, but it might also lead to fibrosis. Okay, so this is the big picture view. I'm going to close. We see that if you have with other infections, you get a robust immune response, affinity maturation. In COVID, you tend not to get, you have lower affinity antibodies, actually lower breadth in particular is also relevant, and you have more CD4-CTLs than CD8 T cells. And that's the big picture. I'm going to thank all the people who did the work in the lab. So most of the tissue work was done by Naoki Kaneko. A lot of the work that I talked about on vaccination came largely from Cory Perugino and Hang Liu. And I had lots of collaborators whom I actually mentioned at the beginning. And I'll stop sharing and take questions if any.
Thank you very much, Professor Kokolay. And it was a really wonderful talk. And we'll be able to take a few questions from the panelist or the attendee. And if you have a questions, you can raise your hand. And we'll be able to address it. We'll be able to convey to the speaker. For those who are attending, they can also put few questions in the question answer box, and we can facilitate conveying this to the speaker. So let me give some time. It's always a bit lag in this. So, yes, I think the first question, I think there are a couple of, the questions coming up. First one, I believe, Dr. Veena Patil. Yes, you can unmute it. You can do it. Okay. No, that's fine. I am not, but unmute her.
Okay. So, am I... Okay, am I audible?
Yeah. There's some feedback. Maybe others should mute. Yeah. Veena, you can go ahead. I think the others need to mute.
Yeah. I think I'm just going to switch here and then ask the question. So it was a wonderful talk. It was so heartening to see that CD4-CTLs are really playing a lot of important role in this. My question is about the B cells. there are a lot of dynamic changes during pre, post, and different doses of vaccination about this CD27 low and high and CD. So my question is like, would there be, have you checked at the BCR level and have you seen that they are sharing the BCRs to be able to say, this is the population, same population, which was expressing 27 is going low on 27?
Yeah, so we've looked a little bit, we did BCR-seq. The number of spike positive cells is kind of low. So we do see some, in humans, especially since the samples are small, we do see some sharing, but it's not going to statistically satisfy anybody there. So we are looking at the total population right now, which is not looking at spike positive only. And there, because we have COVID patients also, we can see the links between cell types. So that's where I think we will show you some links. But most of this is not based, it's based on transcriptome rather than BCR right now.
Okay, my one more next question is, if in the germinal center, if TFH cells are lesser and CD4 CTLs are becoming, is this the TFH cell that is becoming a CTL, CD4 CTL, or? So
we think, and I'll tell you, so I have a graduate student in my lab who's actually studying this in a mouse context. So we think it's the precursor of, so the cells that are in extrafollicular, where you have TH1, TH2, and so on, you have a lot of CXCR5 positive cells. Those, some people have, called them TFH cells. They're not TFH. They don't have BCL6 yet. Those are the cells that drive class switching. It's that pool that is failing to give rise to BCL6 positive cells, but in the presence of TNF, appears to give rise more to CD4 CTLs and TH1s. If you block TNF, you will get back germinal centers and TFH cells. So this is, so it is basically an effect on differentiation. We have some data. We did a detailed analysis at using whole genome bisulfite sequencing, everything else of different populations. And basically what we are seeing is that, yes, there is this pool of cells that seems to divert, which is a little, so that's my guess. I think the data suggests that is correct by the TNF blockade, but I think we still, again, we would like to see connections with BCR-seq showing the, with TCR-seq showing the connection and so on. These are all ongoing. Even the COVID work that I showed you at vaccination, the experiments are still going on right now. Some of the data is from 2 days ago.
It was a wonderful talk. Thank you so much.
Thank you.
All right. I'm going to take it. Yes. Professor Pillai, there are a couple of questions on the question answer board. I probably can pick up one or two. So one is from Vishal Rao. How does the levels of germinal center reactions generated differ between homologous and heterologous boosting?
So heterologous boosting is done fairly infrequently. So what I don't know the answer to that question. I don't think there should be any difference. I actually do think that if you boost after infection, that's what we've looked at, you do get the type of cells we would call the more germ center type cells after boosting in COVID patients. But heterologous boosting with 2 vaccines, we have not looked at. But I would suspect there would be no difference.
Okay, thank you. And I think there is one more question. So I will try to sort of read it out. The question is, what could be the probable mechanism for protection from greater variants in vaccination versus COVID infection, as you mentioned? What could be the mechanism that, yeah.
I think the mechanism based on, there's an interesting paper from Michelle Nussenzweig from 2021. There's a paper that came out recently from Scott Boyd, and there's this work of ours. We would all argue that there is one type of memory cell that comes out of the germinal center which has greater breadth. So at least we are showing it in the memory cell. They have just shown it at the level of antibodies. That vaccination, because you allow cells to be mutated in the germinal center, may allow for a little more diversity of your memory pool, which then allows you to see slightly different forms of the original protein. So the purpose of a somatic hypermutation, I think, is twofold. One purpose is affinity maturation to give you high affinity antibodies coming from plasma cells. The other purpose is to diversify your immune repertoire. The memory cells will become slightly higher affinity, but they will also be broader because they've been mutated and not selected. Remember, outside the germinal center, you don't get any selection. In the germinal center, you got selection, but then you stop selecting and you let them go out. So that might give you more diversity. That's the way we think about it. But again, it's to be proven further.
So yes, I mean, there are some students are, yes, I believe you love to answer them anyway. So one from our NIH, Chandrima Bharadwaj. So he's asking about this, your mechanistic link with exist RNA. And is it the reason she thinks that there could be some sex bias in terms of disease severity?
It could be. So one of the reasons for sex bias that has been proposed, which is that maybe you're protected better if you have two X chromosomes that are functioning, right? If you failure of X inactivation in some women might give them an advantage. That could be a reason for sex bias. That's usually linked to TLR7 in people's minds, but it's not something that has been solidified. Clearly, beyond sex bias, there is a somatic role for exist. And that has come out from a few different cell types, including B cells. And we are now showing, okay, there's one particular type of B cell that this makes complete sense. And it's the only type of B cell we can show which has very high levels. It's a defining transcript in these cells of exist. So, and I think the idea, the effector cells, the precursors we describe as what we think is coming out as a pre-plasmablast-like cell, all have a signature of having been activated through TLRs. So so the TLR7 connection, the induction of Tibet, induction of genes linked to Tibet in the precursor populations might be because of loss of exist. This is at this point, we are just saying that there's a data from a cell line, that's cell line data. There's a data from lupus patients showing you have more of these activated type cells. And then now our data saying, hey, look, there's a specific population which has it, and its potential successor doesn't have it at all.
So, and I think it's probably the last question for today. One of our own, Dr. Nimesh Gupta, he's asking a question. I think he's putting it in the question box. These extrafollicular B cells that you're talking about, are they also getting help from other subsets other than, of course, T follicular helper subsets?
Yeah, so I think we've gone into a bias in the field. The first bias, which was, I think, incorrect, was that all class switching is also driven by T follicular helpers. That is clearly not the case. We showed this in humans in 2017. Carola Vinuesa showed it in mice the next year, that at least we showed for IgGs, switching is happening outside the germinal center. So now, are the cells that are outside the germinal center, we should not call them TFH cells, just it's a definitional thing. It's semantics, right? If it's outside the germinal center and doesn't have BCL6 or very low level of BCL6, I will not call it a TFH cell. So it's a non-TFH cell. Now the question is some classical subset of TH1 cell doing something only in the mouse? In humans, IgG switching is not being driven by interferon gamma, for instance. So we have unique cell types, some that make, we have shown IL-4 and IL-10 producing cells will drive IgG4. Others have shown in the germinal center, IL-4 and IL-13 will drive IgE. So there are different types of cells. Some are true TFH cells driving switching into IgE, but the ones extrafollicularly, we shouldn't call them TFH cells, I think, just for the clarity. We call them pre-GC TFH, probably not a good term, but that's terminology.
So with this, I think let us thank Professor Pillai for such a wonderful talk and experience, and for your time and patiently answering all these questions, sir. And we will probably go to the next speaker of the session. Thank you again. So with this, yes. So let me now, I believe, yes, I think we have the second speaker. Let me welcome Professor Dwayne Wiseman. in this forum, in this NII Symposium on COVID-19. So the ongoing pandemic and this accelerated vaccine development and introduction have created a sort of global scale laboratory for further our understanding of the amplitude and duration of this humoral immune response. Interesting part is this recurrent emergence of variants of concerns. Over the last year or so, along with the presence of vaccinated, partially vaccinated and unvaccinated individual in human population. This led us to realize the importance of the breadth of antibody response and underlying mediators. Today, Dr. Duane Wiseman will discuss these important aspects, comparing vaccine and infection-induced antibody responses. Dr. Wiseman obtained his PhD from University of Alabama School of Medicine, subsequent to a residency at the Brigham and Women's Hospital, Boston, and a postdoctoral tenure at Children's Hospital in Boston. He joined Harvard Med School in 2010. Currently, he's serving as an associate professor. His laboratory uses a plethora of techniques, including mouse genetics, human studies, cellular biology, single-cell transcriptomics, and computation, to elucidate the underlying features and elasticity of antibody recognition capacity. Let's hear from Dr. Wiseman. Please.
Thank you so much. Can you hear me? Okay.
Yes, we can hear you. Yes.
Perfect. Thank you so much. It's really fun to be here and very difficult to follow Shiv's talk. That was excellent. Thank you, Shiv, for that. So yes, I'm going to talk about durability and breadth of antibody responses to SARS-CoV-2 as well as to variants. And so I think it's fair to say that it's a problem that one of the big problems of the pandemic is that there are evolving variants and that immunity can wane. And so some of the questions related to the antibody response that we have is what influences the breadth of recognition and what influences the longevity of secreted antibody responses. Because we know that antibody responses can vary in magnitude as well as composition, in addition to longevity. And as Shiv very nicely illustrated and presented, the memory in the B cell system is housed both in the secreted antibodies that are soluble in the bloodstream, as well as in memory B cells, which express the antibody on their surface. And I have this non-equal sign here just to illustrate that these provide non-redundant functions. And so again, as Shiv illustrated very nicely, the antibodies can come from different kinds of developmental pathways in the B cell system. So a naive B cell can be activated and can produce antibodies through a germinal center response, as well as through pathways outside of germinal center. And as Shiv mentioned, this is an extrafollicular kind of a pathway. And this is just to, in a very simplistic way, review some of the features that are important or that are associated with either an extrafollicular response or a germinal center response. And since Shiv mentioned this already very thoroughly, I'll just review here that an extrafollicular response can produce memory B cells as well as short-lived antibody secreting cells. And the germinal center response is unique in that that's the pathway that allows for greater affinity maturation through somatic hypermutation, which is this SHM thing here, as well as production, we think, of the long-lived plasma cells that I think the thinking is that this is germinal center dependent. And okay, so I think, in medical school training, one of the key teachings, especially in classes like anatomy, is that to understand function, understanding the structure of things will help provide a better understanding of how things work. And so what I'm going to talk about now to begin with is basically the structure of the antibody response with respect to the SARS-CoV spike protein. So in other words, where, like for a human antibody repertoire, how do we recognize the spike protein? What parts of the spike protein are recognized? And how does that influence how the antibodies work? And so this slide is showing you, just reviewing some data that we published a few months ago, where we took individuals that had suffered from COVID-19. These were mostly mild cases that recovered as an outpatient. So these were non-hospitalized. They had symptoms and they recovered. And so we drew blood from these folks about a month after they had their symptom onset. So this is still a very early memory B cell response. We took their memory B cells. This is CD19, CD27 positive, sorry for the typo here, IgG positive and spike positive B cells. So as Shiv mentioned, the memory B cells are very heterogeneous, and he's doing excellent work on that. And this is just the general CD27 positive, IgG positive, spike positive. We cloned the antibodies out of these memory B cells from 19 individuals. And what we wanted to do is just ask, where does it bind on spike? And so to get a general view of this, we did a competition analysis with these monoclonal antibodies. So that's what's shown here on the right side. So the axes, so the y-axis here, this is just names of our monoclonal antibodies that we cloned out. And then on the x-axis, it's the same antibodies listed in the same order. And the difference is that these antibodies, we made two versions of each antibody. One version of the antibody was biotinylated, so we can detect it in an ELISA assay. And then the other antibody was left unbiotinylated, so it would act as a cold competitor. And we did this such that the competitor was 10-fold higher in concentration, so we can capture a range of affinity differences that might occur between the antibodies. And so what happens is when the antibodies block themselves, we color it in this darker color. So as you might expect, antibodies block themselves in terms of the same antibody in competition with the same antibody. That's where the diagonal line comes from. And then we see that other antibodies that are different might compete with each other as well. And so that's where these clusters come from. And so what we find here is that in general, there are only a few major, what we can think of as footprints that antibodies recognize on the spike protein. And some are bigger than others. And this reflects immunodominance. And so, for instance, this big square in green is we mapped it to the RBD region, and we call it RBD1 just because within RBD, it's the most frequently recognized region. And then there's RBD2, which is slightly smaller, and then RBD3, which is again smaller. There's a couple of regions on the N-terminal domain we call NTD1 and NTD2. And then the S2 region has really three regions. So it's we call S2-1 and S2-2. And there's one antibody that doesn't compete with anything. That it binds to the S2 region, but it didn't compete with anything in our panel from these patients. So the heat maps below indicate some of the other features of these antibodies. So the ACE2, for instance, this is the ability of the antibodies to block spike binding to ACE2. And so you could see that most of the activity here maps mainly to RBD-2. as well as some of the other RBDs, and mildly to NTD. And then below are some of the other features in terms of what other segments that the antibodies bind to, as well as other coronaviruses like MERS, HQ1, and OC43. And you can see that by and large, they cluster, like this binding activity clusters with these competition groups. So this is basically just mapping out how things bind. And since the data are published, I'm just going to review some of the general features here. This is where these, we call them epitopic regions, because it's quite a gross generalization of where the antibodies bind, because they can block each other. That doesn't necessarily mean that they bind to the exact same epitope, but they might be binding near each other, or at least influencing each other's binding. And so shown here on the left is a spike protein that's a profile of it, and then looking down on, from say, from north, looking down on the spike protein, you can see the RBD2 and RBD1 and RBD3 are here clustered on the RBD region, and the NTD2s and S2 down here. Okay, so the graphic on the right represents some of the features of these regions. So shown here in these boxes with antibodies inside, this represents the targeting frequency. So the more antibodies there are, it's a relative immunodominance. And these little dials in the middle represents their neutralization capacity, basically. So you can see that RBD2 is the greatest neutralizers, whereas in S2 they don't have as good of a neutralization ability. And then the dial here that's colored in black, this is the cross coronavirus activity. So you can see that for S2, some of the segments of S2 are really great at cross binding other coronaviruses, as you might expect, since this is a conserved region. Whereas the areas in RBD, they're not as good at cross coronavirus binding, but they do vary a little bit. And then the information on the far right shows the escape capabilities, basically, or the resistance to escape by these antibodies. So you can see here, for instance, that RBD2 and NTD1, they're very prone to losing their activity with the variants. And so I wanted to point out too that there are some interesting antibodies that basically have the best of both worlds, where they are broadly reactive to other coronaviruses and have some at least modest neutralization activity. And so here's some representative figures from other papers from the Wiesler group, as well as from Dennis Burton, where they've cloned a very interesting antibody that binds to the very bottom of the S2 region called the stem helix, that has neutralization activity, as well as it can bind across the coronavirus family pretty broadly. And so we took these antibodies, one is called CC40.8 and one is called S2P6, to do our competition analysis with these antibodies. And we found that there was one antibody in our panel that did compete with these. And that's this one down here, S2-3. So that's where these antibodies belong in this competition group. And so I guess, the good news is that there are antibodies that might have the best of both worlds. For instance, you can imagine if there were a vaccine that can elicit these, that it might provide a broad protection, or at least neutralization ability across different coronaviruses. The downside is that, as shown here, it's very infrequent, and the function doesn't necessarily transmit to any antibody in that competition group, as our antibody C12b3 does not have any neutralization ability, and it doesn't bind to other coronaviruses as well. And so this slide is to show you a little more data that includes Omicron, and basically where the neutralization activity resides. And so as we saw before, the RBD2 and RBD1 has some neutralization ability. And so what we did here is we wanted to ask, where the frequency of neutralizers are, and then where the potency activity is. And this will become important later on in the talk when we discuss some issues about mechanism of how boosting might influence breadth. And so here what you see is pseudovirus neutralization activity with the different variants listed, and then a panel of monoclonals that are representative from this group of people that we cloned early after recovery from COVID-19. And so you could see in this teal color, this is basically just yes or no answers regarding whether or not there are neutralization abilities in that cluster. And so you can see here that this group RBD1 has the most frequent, is basically the most dominant place where you can find neutralizers. And the good news is that the neutralizers are, the antibodies in RBD1 are relatively resistant to variants. So you can see, for instance, for Omicron, you still see that you find the antibodies that still have activity for neutralization. However, if you ask the question about potency, RBD2 is the champion here, and RBD2 is more susceptible to viral variant escape from the activity. So frequency of neutralizers and potency, and are basically being separated here to illustrate a point that, again, we'll talk about in a minute. All right, and so as you can see here, there's a few neutralizers that go all the way through Omicron, and the reason for this is that antibodies in RBD3 and RBD1, they bind to the flanks of this RBD region. Shown here is RBD in gray with the Omicron mutations shown in red. And so some antibodies that bind to RBD can avoid some of these mutations, whereas antibodies in RBD2 are mostly lost. There are rare instances, for instance, this C98C7 that does retain binding, even though it is a part of this competition group of RBD2, but most of them are lost. All right, so just for the last part of the talk, I'm going to switch gears a little bit and talk about durability of secreted antibody responses. And so what we did here is this green cohort, which had mild COVID-19. This is the cohort that we were just discussing, where we cloned memory B cells early after. So we continued to follow these people over time and compared them to individuals that had just gotten the vaccine without having COVID, and that's shown in black. And then the purple group, these are folks that have recovered from COVID and then got the vaccination. And so these are data that are run out over 200 days, showing the trajectory of the secreted antibody responses. And so on the left is anti-S IgG and anti-RBD IgG on the right. And so you can see here that recovery from mild COVID, you have very modest antibody responses, but they tend to stay relatively stable compared to, say, someone who received 2 doses of mRNA vaccination in the black line, which tends to start much higher, but then they decay at least to the level, maybe even below people that recovered from infection. And then the purple group has magnitude as well as a less of a decay slope. With regard to variants, as many people have shown, Beta and Omicron are more resistant to, or they are better at escaping antibody responses. And as you can see here, is shown after 2 doses of mRNA vaccination. The same color scheme applies here. The green group, this is just COVID early and late after infection, and then the vaccination after infection in purple and vaccination alone in black. What we're also showing here is what happens after the third dose. And so particularly over here in Omicron, which is similar to beta, is Omicron escapes the antibodies after infection pretty well. Vaccination after infection, you have a much better recognition over time, but vaccination with 2 doses alone, you get almost nothing to Omicron. However, a third dose of the same vaccine, an mRNA vaccine, gives you a much broader recognition. It gives you Omicron recognition that is not specific to Omicron. You also see it to beta and delta as well. And so this is basically the same data with just the variants plotted out this way. So you can see and directly compare the neutralization activity across the different variants. And that basically what happens is the third dose evens out cross-variant recognition from the antibody response to levels similar to people that have recovered from COVID and gotten 2 doses or 3 actually of vaccination. Okay, so what is the mechanism of how this happens? So what I'm showing here is just illustrations of magnitude and content. And so what I'm talking here about win by magnitude is basically if you have an antibody response illustrated by this small rectangle here that has its composition that I'm illustrating here by just this red and yellow, where red might represent antibodies with cross-variant recognition and yellow might be more variant specific. And so if you make more of what you have and you keep the composition the same, you can still win across variants. So you have more red because you have more of everything. There's also win by content, which is you can have a very similar magnitude of antibodies that recognize the virus, and you could have more of the red within the same group, or you can do both. And so, as illustrated by these plots, it suggests that there's actually a content thing going on here. And what we are interested in seeing if we can measure this a little more carefully with the content issue. And so we did this by just calculating an index, and I'm not going to actually go over this because time is running short. But the conclusion is basically that the third dose evens out the recognition over different variants. And so Shiv talked about this in his talk and gave a very nice, hypothesis of how this can work. And in addition to that, we think that one thing that might be contributing is illustrated here. And so as Shiv mentioned, the somatic mutation and infinity maturation occurs, and several labs have shown that this occurs over many months. And so this is represented here by this red triangle thing here at the bottom. So this is the affinity maturation and somatic mutation that occurs over many months. Between the first and second dose, there's not a lot of time for that to happen. At 1 month after infection or vaccination, somatic mutation is very low. Whereas if you look 3 to 6 months after, it's much higher. And that correlates with affinity of the antibodies. And these boxes here represent the epitopic regions. And the color, red, orange, or yellow, represents basically the potency of neutralization ability from the antibodies. And so what could explain, or at least contribute, we need to figure this out and whether this is true or not, but one of the ideas is that what could change is just the color of this middle box. So you have areas that are potent, mild, and weak to no neutralization. And with affinity maturation, the orange box can turn to red. And so what I'm thinking of here is this RBD1 region, for instance, as an example. We know that RBD1 contains lots of neutralizers that retain recognition already to the variants like Omicron. If you boost the memory B cells that have had time to mature, like for instance in this RBD region, then that could result in more potent neutralization. All right, so I'm going to skip that, and just in the last minute or so, I'm going to explain a little bit. So Shiv mentioned this idea about the sustainers and decayers, and we think that sustainers probably is evidence of more germinal center activity, which led to more long-lived plasma cell responses. So what we found subsequently is that in this sustainer population that had evidence of higher somatic mutation in a bimodal kind of a distribution early after. So as I mentioned, most people, the memory B cells don't have a lot of somatic mutation, but in sustainers, there was a small group of memory B cells that had high mutation. And these memory B cells ended up binding more to the S2 region as well as cross coronavirus recognition. So this seemed to be a signature to maybe predict who might have longer term antibody responses. And this correlated with cross variant recognition in sustainers compared to decayers due to magnitude, because the content of this antibody response between the two at the same time was very similar. But sustainers, because they held on to more antibodies, had more cross-variant recognition. So in summary, SARS-CoV-2 variants escape the most potent baseline neutralizing antibodies, but the moderate neutralizing antibodies targeting relatively immunodominant RBD regions are more resistant to escape, and we think these might be, at least in part, responsible for the greater breadth after boosting. So broadly neutralizing stem loop target is very rarely targeted, although this would be a nice thing to be able to elicit in a vaccine. It might be very hard to do that. And then high memory B cell cross coronavirus reactivity is an early marker for longer lasting antibody responses in those where we can detect the memory B cells might have been recalled. That's connected also to a more durable response. And then last thing here, the third dose leads to increased evenness of cross variant neutralizing function. And so the data that I showed you, mostly done by Yuezhou Chen and Pei Tong, with a lot of help from Adam Zoyani, Avnish Gautam, and Noah and Ali, as well as a lot of help from collaborators. And so thank you so much for your attention.
So first, let me thank Professor Duane Weisman. It's a wonderful talk. I mean, this whole area of carefully navigating this quantitative and qualitative attributes of durability of breadth of antibody responses. A very interesting talk, and we do certainly have several questions. First, we will start with Professor Pillai himself. Yes, yeah.
I don't have a question. I can save my time. I can talk to him all the time. So I'll save my time for this.
It will be okay. I think we can probably also learn from this.
When we should talk offline, I think when you take 27 positive cells, you're in those, your sustainers, I think you're starting with people with that common cold coronavirus, cross reactivity, which is present in both 27 positive, 27 negative. And then as you activate, with disease or vaccination, lots of them are entering that effector 27 positive pool, or what I'm calling precursor 27 positive pool. So when you take 27 positive, I think you get a mixture of things that have come out of the germinal center and things that may not have, but they're older cells. So there's a chance that they're more mutated maybe. And that might be why you have what you see. But I could have always told you this offline.
Yeah, no, it is really interesting. That's probably, I totally agree. I think there's probably evidence of them going, or at least it probably means that they're going in the germinal center. And what's interesting too is that the longer lived antibody responses include RBD reactive as well as the S reactive. So it seems like they're, in addition to recalled re-entry, it seems like there's something else that's going on to provide an environment that also allows, like the RBDs that aren't cross-reactive to, like maybe the new people, the new guys coming in also are also benefited by that environment or something. I don't know.
So I think part of that might be the delay that you're seeing. We all see the delay, right? That delay may be that the RBD naive cells, mainly naive, have to enter a germinal center and then get mutated, and then they start contributing more to neutralization than anything else. So maybe the part of the delay is RBD being brought in, and S2 is there earlier, but who knows? Like, that's interesting.
Yeah, there's something going on, something interesting going on.
Yeah.
So thank you. And I think we will have the second question from Professor Dr. Giridhari Lal. He's from National Center for Cell Science, Pune. And he has a question for Professor Wiseman. Giri, you can go ahead.
Okay, thank you. Fantastic talk and excellent work to show that how different region of your RBD domain generating then different clones and neutralizing potency. So my question to you, if you look at mucosal associated viruses, when they have entry point, IgA is the one which protects them. So in terms of vaccine response, have you looked at the generation of any antigen specific IgA antibody in the system?
So yeah, I love the question because we are also very interested in mucosal antibody responses. In this analysis that we did, we have not looked at IgA in the mucosal surfaces. We were able to draw blood from the participants that we're collecting, and so we did not collect nasal specimens or any other mucosal specimens, but that would be very interesting to look at. This was early on in the pandemic in March of 2020, when even the clinical centers that collect these were actually all shut down, at least for the outpatient side. And there were folks that were on the inpatient side collecting these, which are great friends and collaborators that I think have looked at this and are generating great data about this. But for what we did was we just created our own cohort early on, and we were limited because of the biosafety concerns to get people that had recovered. And so we didn't get mucosal samples for these folks. But it is an interesting question of what's going on at the tissue barriers and what kinds of antibody responses are there. We don't have information on that with this cohort.
Perfect. Thank you very much. So can I ask one more question?
Yes.
Looking at the various clones, what you have and their neutralization potential, have you looked at the class or subclass of the IgG response or IgG type they are generating and their neutralization potential?
You mean like IgG 1, 2, 3, or 4?
Correct.
Yeah, so we used a common primer that is able to capture all of them. And so we don't, unfortunately, we aren't able to really distinguish those. Look at that again. We were talking about this before. There may be some very minor differences between a couple. We might be able to get some information, but right now we just, they're a mix of all of them.
Okay, all right. Thank you very much.
Yeah.
So I think we have some time, so you can probably continue with some questions. Some are coming from students and other participants. One question went that, is this escape solely dependent on the mutations in epitopes recognized by the neutralizing antibodies, or there are other factors that you might suspect could be involved too?
Yeah, great question. As far as I understand, there's, and in our data, the escape from antibodies correlates very well with the position of the mutations. So, although there may be other reasons and other ways that this happens, but there seems to be a very, very tight correlation with where the mutations are and where the antibodies then are affected. Let's see, I think you're mutated or muted. I meant muted.
Yes, getting there. If we continue with this web platform for a few more years, as a scientist, we will get there soon. So I think. So I think another questions. I think there is a couple of more questions. So one, I think Sandeep Jawan, he's asking that which chain is involved in binding spike protein in which CDR region?
Oh, yeah. So we are writing another paper. So Steve Harrison is doing some analysis here with our antibody panel to describe a little bit of the heterogeneity of where on the antibody, basically the paratope, the part of the antibody that is kind of the corollary to the epitope. So there are, some of these early antibodies that came out that were high affinity and good binders, good neutralizers that use VH3-53, for instance. They tend to use the VH encoded, sort of the germline encoded CDR regions, so CDR1 and 2, to interact with their targets. Whereas other antibodies use, different, parts like CDR3, as you might expect, as well as there's other kinds of antibody clusters that might use more light chain contribution. And so there's a diversity of how the antibodies work, and with everything, and then with the spike protein, it's no exception. There's, the antibodies figure out a way to kind of interact with it via all of its tools, so there's a diversity of it. I think you're muted again, or mutated.
So this is one more question from Vishal Rao. So the question is about, he's projecting a scenario that people with severe COVID-19 from the first wave would they show lower recognition to RBD2 compared to mild individuals, since the protectivity of RBD2 offers in terms of neutralizing capacity places it under higher immune pressure? What sort of, conjecture?
Yeah. So whether or not the severe folks, whether or not less recognition of the more potent region. My guess is no. My guess is that they probably have made good antibodies to all the parts, but they just made them too late, or that the severity caught hold and caused the disease. Because we know that severe disease can generate larger antibody responses. But I think it's just, not directly related, but, it's just that they weren't there in time, that I think. But that's just some guessing.
Okay. So, and I think we have one question from Arvind Sahu. Just let me check. Yes, he has been unmuted. So, Arvind, you can go ahead. Arvind Sahu from National Center for Cell Sciences, Pune, India. Arvind, can you hear us?
Okay, if he doesn't join, I just have a quick question before your
Yes, Sheep, please go ahead. Please go ahead. Yes.
I noticed Anna's comment on IgA. So to my knowledge, there's some people have reported IgA after vaccination, but in reality, you shouldn't get secretory IgA from a parenteral vaccination. So we don't expect it. You may see in blood some IgA, but whether it'll be mucosal IgA, I don't think there's good proof of it. Though interestingly, there is some IgA that has been reported after vaccination, though it's not high. And maybe Alex can address that in his talk, which comes next. Yeah. So Anna has a hand up. Yeah. Anna, I can't unmute you, unfortunately.
Anna? Yeah, we have to unmute you. Just, yeah, it's a bit complicated, but we cannot hear you. We have to unmute. You have to unmute yourself.
It was just a comment because Dwayne mentioned that he had only blood samples and not nasal swabs. So given that, I thought if he's interested in IgA, he might want to look at that. That's it. I mean, I agree with you. I don't think you're going to get a whole lot of IgA. Yeah.
Yeah. Thanks. Good to see you, Anna. Yeah.
Yeah, we looked early on, and so we didn't complete the whole longitudinal analysis, but we did find that IgA from the blood was actually decaying quite rapidly. But
am I audible?
Yes, Arvind. Okay.
Yeah, am I audible?
Yes, I think we'll move. I think let us thank Professor Edwin Wisman. Oh, Arvind. Okay, all right. Sorry. Yeah, just last question for Edwin, I guess. Yeah, Arvind could finally unmute
sorry,
it.
I had trouble with this. So
yes, fine. Yeah, please go ahead,
thank
Arvind.
you very much, Professor Duran. It's a great talk and nice to see my student Avnish working there and trying to do hard work and seems to be involved in multiple work. My question is to you is that after the third dose of vaccine, are there any ways, markers which you think which would allow to predict how long the memory is going to last? Hello?
Duan, you are muted. You are mute. I think this is not working. We have to have that computer here. Yes, Duan, you are unmute.
okay, now I'm yeah. I was clicking
sorry,
on it, but then it wouldn't unmute me. So
Anyway,
so it's a great question, and Alex is going to talk more about the third dose, and maybe he has more insights on that in terms of signatures. The only signature we have is long-term antibody responses seem to have correlated with early mutated memory B cells in the sustainers. But in terms of a third dose, people seem to be able to be homogeneous, at least in our small cohort. People kind of go up together, and how that might distribute over time, we don't know yet, but Alex might know more about that.
so I think with this, let us thank Professor Duan Wisman for this fantastic talk and this and his time, of course, and this very lively discussions after this his own talk. Thank you very much, and we'll move to the next speaker. So the next speaker for this session is Professor Alejandro Valazis. It gives me immense pleasure to introduce him in front of you. He is an assistant professor of medicine at Harvard Medical School and a principal investigator at the Reagan Institute of MGH, MIT, and Harvard in Cambridge. Dr. Valazis has received his PhD from Harvard University and conducted postdoctoral research at the California Institute of Technology prior to joining the faculty at Harvard Medical School in 2014. He leads a laboratory that explores the fundamental mechanism by which the immune system prevents the establishment of infection by employing various immunological engineering tools to dissect the underpinning of the protections. And today he'll be again talking about humoral immunity to SARS-CoV-2, particularly with some emphasis on neutralization potency of humoral immunity and what could be boosting the antibody response rate. So going into the topic, Professor Valazis, with this, I welcome you in the forum.
Thank you so much for that kind introduction, and thank you to the organizers for giving me an opportunity to tell you all a little bit about some of the work we've been doing in our lab to understand antibody responses to SARS-CoV-2 and its variants. So I know this has probably been covered in all of the previous talks, but just to make sure we're exactly on the same page, when the virus infects a susceptible host, what we observe is obviously the innate immune system functioned first, just prior to sort of the onset of symptoms that are observed in the patients. And within just a few days, you have the priming of a T cell response, which can then help prime the B cell responses, which go on to generate all of these antibodies that I'll be talking to you about in the talk today. And so those are primarily in the form of initially IgM responses, which ultimately wane. and then followed by IgG responses, which are significantly more sustained, as well as class switching to IgA responses, which came up, I think, in one of the questions there. And so with this kind of as a framework, we were very interested in understanding, how antibodies correlated with the range of severity of disease that is observed for SARS-CoV-2. So as you all are likely aware, COVID can present in a number of different severities, including asymptomatic cases and those who have very mild disease, which tend to occur in younger individuals, as well as much more severe cases of COVID, which tended to occur in individuals of a more advanced age, and very, very severe and occasionally lethal cases of COVID, which largely occurred in those who were above the age of 68. So really, there's this range of different experiences of people who become infected by COVID, and we wanted to understand how antibodies and neutralizing antibodies in particular fit into this range of disease. And so I'm going to tell you about the work that we did, and this is all. I should maybe start by thanking Wilfredo Garcia Beltran, who was the first author of this and all of our other papers on COVID. He is an amazing physician scientist at MGH, who is now has his own lab at the Reagan Institute, and I must admit actually gave me many of the slides that I've used in this talk. So he really has had an outsized influence on all of this work, and he'll be acknowledged even further in the end. But this was a study that we undertook at the Mass General Hospital, looking at individuals of varying disease severities and trying to understand how antibodies fit into that process. And so what we did was recruited individuals who fell into the following cohorts: those who had COVID, confirmed COVID cases, but who were not hospitalized; those who had confirmed COVID cases who ended up in the hospital being admitted. Those who were admitted, there was a group that had become intubated, so because their disease became too severe, as well as a cohort of individuals who ultimately passed away of COVID. And we separated out those who were COVID infected, but who were receiving immunosuppressive drugs for other reasons. But just looking at some of the demographics here, what you can see immediately is we're replicating, what many others have seen, which was that the younger individuals tended to be the ones who didn't have to go to the hospital. Interestingly, we, as others have seen, observed that those most severely affected were men, and that women tended to fare better in this disease. And then looking at the number of days by which the patient was positive. So for 11 days, or you would have much longer courses of infection, sort of the more severe you became. But interestingly, those who were immunosuppressed had some of the longest PCR positive results, which I think others have argued may be contributing to some of the variants that we see emerging later on in the course of this pandemic. But interestingly, those same individuals who were immunosuppressed actually spent some of the fewest days in the hospital. So the immune system really is sort of contributing to the pathology and to the need for hospitalization. We also observed, as others had seen, that comorbidities and having pre-existing medical conditions sort of made you more and more susceptible to having a severe course of infection. So the risk factors, as others have already mentioned before, were age, male sex, and comorbidities. And so we wanted to understand, how this played into what we were observing at MGH. And so if we look at the levels of an inflammatory marker, C-reactive protein, we actually see that those who had to be hospitalized had significantly higher levels of inflammation in general, as well as significantly increased levels of IL-6. The more severe disease had higher levels of IL-6. And this was at the same time that lymphopenia was more often observed in those who were more severely ill. So it appeared that disease severity actually correlated with these inflammatory markers in lymphopenia. And so before I get into the antibodies, just to make sure we're exactly on the same page, I'm sure this has been already gone over in other talks, but the particle actually contains many proteins that could be recognized by antibody responses, including spike, membrane proteins, envelope proteins, and the nucleocapsid that's internal to the particle surrounding the single-stranded RNA genome. And we wanted to explore the range of antibody responses that were occurring to these particles. And of course, the spike is what interacts with the ACE2 receptor on the surface of epithelial cells, which initiates these infections. And more specifically, this RBD domain, the receptor binding domain, is what engages ACE2 and what can be blocked by a neutralizing antibody. In many cases, they're against the RBD. So the reason I say that is that at MGH and at the Reagan, we developed an ELISA early on in the pandemic to measure antibody responses that could detect antibodies that had arisen in patients against both the RBD region or the larger spike. And that's based on simply coating ELISA plates with those two purified proteins, and then allowing sera to bind and detecting with either IgG, IgM, or IgA secondary reagents, such that we could measure independently these different antibody responses that were present. And so we did this for our individuals who were in these different disease cohorts, which I have already told you about. And what we observed was the following. So IgM responses tended to occur within a matter of, 2 weeks or so. Those, they peaked probably in the 3-week range and then it essentially came down. Whereas IgG responses actually, we never really got to a peak in this particular cohort, but they were much more sustained. IgA responses were at a lower level, but more similar to IgM, where they sort of came back down towards the end. And so if we compare this in our different cohorts, what you observe is that in particular, IgG responses were significantly higher. These are just binding antibodies, were significantly higher in patients who had to be hospitalized or had to be intubated than those who were not hospitalized or our healthy control populations who didn't really have these antibodies. And so it appeared that the magnitude of the antibody response itself was correlating to the severity of the disease that was coming up in these individuals. But we wanted to understand the role of neutralizing antibodies in particular in this process. And so my lab developed a neutralizing antibody assay based on pseudovirus, similar to many other labs, in which we take the gene for the spike, the SARS-CoV-2 spike, and co-transfect it with genes for a lentiviral vector encoding luciferase and other markers into producer cells, which then allow us to create pseudoviruses which contain the SARS-CoV-2 spike on their surface. These pseudoviruses can then be used to infect ACE2 expressing target cells, and we can measure the degree of infection using this luminescent reporter in the cells. And that gets read out, and we can determine the dilution of patient sera that enables us to inhibit 50% of the activity of this process. And that NT50 value then is a correlate of the potency of infection, of neutralization by that serum. So we did this for the various cohorts that we had identified, and largely the result tracked with IgG responses. So we actually observed that neutralization in general comes up after a week or two and continues to go up and is sustained for several months. But interestingly, we saw that the neutralization actually was higher in those who had become hospitalized or intubated as compared to those who did not need to be hospitalized. And this is the magnitude of neutralization. So it seems that neutralization itself correlates with disease severity. But one thing that was interesting was that when we compared the amount of antibody being made, so that's the anti-RBD IgG, this is by ELISA, to the neutralization values that we were seeing in our assay, most of these values fell along a very predictable line for most of the patients. But what was interesting was if you look here in red, these dots that are sort of straying from this otherwise very consistent sloped line, and those were in the patients who were either intubated or deceased, suggesting that they were making more antibody, but not getting as much neutralization out of that increased antibody. And so we created this neutralization potency index in which we divided the ability to neutralize and normalized it by the amount of antibody present. And what we observed was that those who had more severe disease or who were deceased actually had substantially lower potency in their antibody responses. And what was interesting was if we use potency as a predictor, we could actually separate out individuals who would go on to have more severe disease or who actually passed away of COVID from those who had a milder course of disease. And so it seems that the potency of antibody actually could predict the survival of individuals. So just to summarize this paper, essentially we observed that having more severe disease correlated with larger amounts of total antibodies, but that neutralization did not increase at the same rate as the number of antibodies present, such that the potency of antibody responses in individuals who went on to have more severe outcomes was lower. And so the take home here is that neutralizing antibodies may not merely be able to help you very much once you have this infection established, but they may actually protect you from future infections. And that is what we started looking at with our studies of vaccine responses, particularly against some of these variants of concern. And so this data is in the last two papers here, in which we were looking at the potential for variants that were arising to escape from antibody responses that were being elicited by vaccines that were being distributed here in the United States. And so just to give you all a little background, early in the pandemic, most of the viruses were derived from the initial Wuhan strain, perhaps with a single mutation in it. But this actually changed with the emergence of the Alpha variant, which emerged in the UK called B.1.1.7. And ultimately, many other sort of variants began to emerge in different parts of the world, including in particular, one I want to focus on in South Africa called the Beta variant, B.1.351. And I'll show you why in a minute, which is if you compare the variants that were emerging at this time, one of the things that was noted was there were a significant number of mutations arising. And particularly, a lot of these mutations were being concentrated here in the receptor binding domain, which, as I was pointing out earlier, is a potential site where neutralizing antibodies can actually inhibit infection. And so we wanted to understand the role of these mutations in preventing antibody responses that were elicited by vaccines from neutralizing the virus. And so we obtained sera from a cohort or several cohorts here of individuals who had received either 1 or 2 doses of the Pfizer or 1 or 2 doses of the Moderna mRNA vaccines and applied them back to our assay, this time modifying the spike protein to generate all spike variants that represented all of those different strains that I just told you about. And what we were sort of surprised to see was that the amount of, this is relative to the ability to neutralize the wild type variant. What we observed was that for some of these variants, in particular the beta variants, there was significant escape from even 2 doses of Pfizer or Moderna vaccine, such that the antibody responses were much less capable of neutralizing them. In fact, very similar to their ability to neutralize unrelated or distantly related coronaviruses, including SARS and WIV1, which is a pre-pandemic virus that has been observed in bats. And so it seemed that these vaccine responses might not actually be able to help us as these more advanced variants emerged. And we wanted to delve a little deeper into why that was. And so we generated a series of mutants in which we reverted the mutations that were observed in the RBD region only back to the wild type and tried to ask the question, to what degree are the RBD mutations responsible for this escape relative to those mutations that are not in the RBD? And this is just to explain that there were actually 3 sort of subvariants already of the beta variant when we began this study. And so we wanted to represent all 3, which had different combinations of these mutations, essentially. And so what is this contribution by RBD relative to non-RBD? And so when you look at the neutralization potency here, what you see is that against the beta variant 1, you have very low neutralization. However, if you revert the RBD mutations back to the wild type, you actually see very significant restoration of the ability to neutralize this variant. And the same was true for version 2 and version 3. And in fact, if we made a variant that only contained those 3 RBD mutations, but none of the other mutations, we recapitulated most of the escape just in those RBD, which is telling us that those 2 vaccine doses are likely making a very RBD-directed immune response that can largely be escaped by these specific mutations. And so what was interesting about this finding was in the context of what emerged last November, and that is the Omicron variant. So, since the beta variant had emerged and everyone was very concerned about it, it turned out that another variant had actually taken over called Delta. And it was extremely rapidly taking over from the original Alpha variant in the UK. But when Omicron emerged in late 2021, it began taking over everything that had been infected with, or had been everywhere that Delta had been spreading, began spreading Omicron instead, including here in the United States, where there was really a remarkable sort of switch between Delta and Omicron over a period of only a few weeks. And obviously, people were very concerned about Omicron in particular, because as you've probably heard already, Omicron actually had a significant number of mutations, so up to 36 different mutations as compared to the wild type spike protein, including, in this case, not just 3 mutations, but 15 mutations in the RBD mutation, in the RBD region, which I just showed you. Even 3 mutations is enough to sort of escape from 2 doses of mRNA vaccine. So we wanted to understand how this might escape from vaccines. And by this time, here in the United States, people had been receiving booster vaccinations. So we wanted to understand whether boosters might have a different effect on these different variants. And so we obtained these different serum samples from people who had received Moderna, Pfizer, and in this case included some Janssen people, either with or without receiving a booster dose. In the case of Janssen, it was an mRNA booster that we were studying. And here from Moderna and Pfizer, it was a 3rd dose of mRNA. It wasn't always the exact same one, and you'll see by the color coding that occasionally Moderna folks received a Pfizer boost, but we didn't see that that made a huge difference. So the results of our study were effectively that if you had been recently vaccinated with 2 doses of mRNA, your ability to recognize Omicron was 43-fold lower, to neutralize Omicron is 43-fold lower than it was against the wild type. If you had received your vaccine 6 to 12 months before, the pattern was very similar, but the magnitude was different in that antibody responses had waned a bit, such that your ability to recognize this Omicron variant was almost none. However, if you had signs of previous infection, and we verified that using a nucleocapsid-based ELISA, so those individuals who had been infected at some point, either prior to their 2 vaccine doses or after their 2 vaccine doses, that actually restored some ability to neutralize Omicron. However, being recently boosted by a 3rd dose of mRNA actually restored a significant amount of neutralization, even against Omicron, which has all of these different mutations. And this was largely true no matter which vaccine you looked at. So in this case, we're looking at the ability to neutralize the wild type variant. We see that people who received 2 or 3 doses of vaccine had pretty similar levels of response. There maybe is a bit of a boost here in terms of ability to recognize the original wild type variant. For Pfizer, there was almost no difference between having 2 vaccines recently or a 3rd vaccine. So it really just boosted your level back to where it was. And in the case of Janssen, having an mRNA vaccine was very helpful. However, against Omicron, the picture was very different. So there was a massive increase in the ability to recognize Omicron with a boost. In case of Pfizer, it was even more dramatic. So this is despite not really changing the level of neutralization against the wild type, your ability to recognize Omicron was now substantially higher. So it seems that a 3rd dose of mRNA actually provides you with a substantial increase in this Omicron cross-neutralizing capacity. So one question that we also tried to address in our study, although I should admit it was very preliminary at the time, but people were really looking for any kind of clues, was why Omicron might have taken over even more than Delta. And so one thing that we noticed in our pseudoviruses was that the titers of pseudovirus that we were making when we put the Omicron spike on them were significantly higher than even Delta or our wild type virus. And if we normalized our viruses to the actual copy number, we actually observed that for any given copy number, we were seeing greater numbers of infected cells for Omicron versus Delta or wild type. And interestingly, what we also found was that in the case of beta, we actually needed more virus to get any given level of infection. And so what we were observing was perhaps helping explain some of the differences where Omicron was actually about 4-fold more efficient at infecting cells than wild type, and about 2-fold more efficient than Delta. And interestingly, beta was actually less efficient than wild type. And this perhaps helps explain why beta, despite being quite evasive from these vaccine responses, never really took over globally. And so this was actually really in line with what others had observed. So after this point, other studies actually came out showing that there were actually differences in tropism and even in the modes of entry by these cells. So there was an early study by a group in Hong Kong demonstrating that SARS-CoV-2 actually seemed to infect bronchial tissue better than lung tissue, and that another group showing that perhaps this was happening using these endosomal routes of entry. So just to conclude, what we found effectively was that antibody responses against SARS-CoV-2 may actually be a double-edged sword in some cases. So we actually saw that low neutralization potency antibodies seem to correlate with having a more severe course of disease, whereas having highly potently neutralizing antibodies actually correlating with a milder course of disease. When variants arose, it seemed that they could actually escape from the antibodies being induced by a standard 2-dose regimen of vaccination. However, that escape was almost entirely mediated by mutations in the RBD region of the spike. But when we saw, when Omicron actually was tested against sera from patients who had received boosting, we actually saw that boosting restored the ability to recognize Omicron, suggesting that perhaps now RBD is not the only region being targeted or the majority of what's being targeted in the case of vaccinations with a third boost. And we also observed that the Omicron spike itself could be significantly more infectious than other variants that we tested. So I just want to make sure I have an opportunity to thank the entire lab. All of these neutralization assays are being run by a wonderful team of technicians who are all going on to graduate in medical school this year, including Evan Lamb, Carrie St. Dennis, and Megan Sheehan. And they've been very much helped by Adam Nitido, who actually used to work on coronaviruses in Wuhan as a Fulbright scholar, and Alex Roeder, a recent graduate student in my lab. But I really have to thank Wilfredo Garcia Beltran, as I mentioned early in the study. He really was the driving force behind almost all of what you've seen here. But I want to also make sure I thank those other labs at the Reagan Institute and beyond who helped us, including Aaron's lab, who provided all of the proteins that you saw in those ELISAs and all of our standards, our collaborators in Germany, including Angelique. And of course, our collaborators at the Mass General, who did amazing work in the pathology department, including Vivek Nananbai and John Iafredi. And of course, the city of Chelsea and the blood donors who donated all of the samples for everything that you've seen here. And with that, I'm happy to take any questions that you may have.
So I think this is a wonderful talk, and I will probably, I think Professor Dr. Pushkar Sharma, I think he's here in this room. He'll be asking the first question, and then we'll pick up a few other questions from the gallery.
Okay.
So it was a nice talk. Just a very quick question. What is the reason for lower potency of antibodies?
The reason that people make low potency antibody?
Yeah,
that's a great question. I think, whether because they have runaway disease, they have a significantly higher level of antigen, and perhaps then they're just making more total antibody, but why they make less neutralizing antibody, it's a little unclear right now if that's the chicken or the egg. Is it that having the disease gives you these low potency antibodies, or is it because you have low potency antibodies that you develop more severe disease? We don't really know if the antibodies are simply a biomarker or if they're actually involved in that process. But I think it's an interesting area to ask.
If I can quickly ask one question. I mean, of course, this is a burning question regarding why Omicron is so evasive. I mean, how can it find the escape route and so on? And then, of course, and at the same time, be more infectious. So are there alternate receptors, possible for Omicron, although the study you showed was basically on ACE2 dependent infectivity of Omicron coding pseudoviruses. So
yeah, I think that is a very interesting question. And, I think we've seen a very, very, I would say very unsubstantiated piece of evidence, which is that in the case of Omicron, we always test our viruses against non-ACE2 expressing 293 T cells, and Omicron pseudovirus was the only one that we saw one dot of infection, even in the absence of ACE2, which I think suggests there could be alternative receptors or perhaps other ways that don't necessarily require ACE2 to enter the cell. I don't want to speculate too much because it's possible that was just contamination or something. But it is interesting, and it would fit that might be why it's actually spreading more easily.
Thanks very much. Although endocytosis-based uptake or entries is interesting thought. Thanks very much for a very nice talk.
Sure, thank you.
So yes, we may have a few questions from the chat box I can see, and also the question answer sessions. Let's pick up one from the question answer session. So again, Vishal Rao. So the question is various markers of disease severity, such as lower in neutralizing potency, high IL-6, T cell exhaustion. Do they generally correlate with each other when it is talking about really a serious disease, or can one of these markers independently influence severity? What is it? Because here also you have articulated neutralizing potency of antibodies vis-a-vis antibody titer, right? So if you can shed some light on it.
Yeah, I don't know if I have a great answer. I mean, I think in general they were correlated, but everyone's course of disease is a little bit different, and so if you look at any one individual, it can be hard to make generalized statements. I think overall things correlated quite well. All of these things correlated well. They tended to have high IL-6, high CRP, low levels of lymphocytes in the blood, and in general created less potent antibodies when they were more severe. But I'm sure you can find individual dots within our plots that those things do not correlate perfectly. So I wouldn't say it's 100% of the time.
So, and I have a quick question for you also. I guess this is, even last talk we've been hearing this antibody report share and, the breadth of the humoral response. And here also we get to the same point that you have boosting, you get to sort of neutralize Omicron. And we can, the question here is this, we're merely talking about separation of these boosting doses, and you are not exactly talking about number of doses. What would matter to actually influence qualitatively the breadth of the humoral immunity, or you actually need to also quantitatively simultaneously influence it by number of doses? I mean, Dwayne was talking about it. He touched upon this. I mean, what is your thought on this?
I think it's a great question, and I hope we can answer it with because if I recall early in the pandemic, there wasn't enough vaccine to go around, and in some places there were people who received their 2 doses of vaccine with more separation. So maybe there's a cohort out there of samples that would help answer this question. I think it would be a fascinating question if we had simply spaced out our doses. Would we have gotten a more broad response just by doing that? Or do we really need a 3rd dose to broaden? I hope someone will actually take that up and answer that question.
If I may, I believe a little more. Some of those animal studies, if we keep this SARS coronavirus aside, might have given some answers earlier, isn't it?
Yeah, I think that certainly there are results in animals that probably suggest that spacing it out more would be useful. But I don't know, whether that'd be exactly the same in a human. I never trust the animal studies to tell us what will happen in the clinical study, because the immune responses can be quite different. But yeah, I think my personal guess is, the more antigen exposure over the longer period of time you have, the more opportunities you give for broad antibody responses to emerge. But yeah, do you require a 3rd dose? I think that's a great question.
Thank you very much. And we have one more last question. One of my colleague, Dr. Gopalan, probably he'll be sitting here and asking this.
Sure.
Hey, very nice presentation. Enjoyed it very much.
Thank you.
Just wondering about this moderate response versus potently neutralizing antibodies by different people for the first vaccine, also subsequent vaccination. I was wondering about the quality of antigen that's produced post-parenteral vaccination. Would that make a difference?
I mean, so are you saying because the mRNA is different than the J&J vaccines? Or what do you mean by quality, I guess?
Yeah, the mRNA has to make a spike protein at the end of the day so that antibodies can be made. Are there a differential between individuals on the quality of spike protein that's synthesized after parenteral vaccination?
It's an interesting idea, actually, not one that I had considered, but yeah, I mean, I guess it's possible that depending on the individual themselves and how they respond to translating that mRNA, perhaps they would make either more or less depending on their own health status. And maybe that does contribute. I mean, certainly we see differences in the quality, in the levels of immune responses generated by different individuals. And if they're very elderly, they tend to not respond as well. But that's complicated, obviously, by the loss of thymus. And I mean, so it can be difficult to tease out if it's because the antigen or because it's the the immune system of that individual. But an interesting point to make.
Yeah, correct. The translation seems to be taken as for granted.
For sure. Yeah, sure. Definitely. Yeah. Thank you.
Thanks. Thank you so much. Thanks.
Yeah.
So I guess, it was, I think it's nice to hear this conversation after the talk. I believe there would be a few more, one or two questions we can still accommodate. We have some time. Nimesh, Dr. Nimesh Gupta, can you unmute yourself and just...
Yeah, hi.
Yes.
Thanks so much. Thanks so much, Alex. It was a great talk. Just have a question on these two types of antibodies. What we see, somewhere we have a lot of binding antibodies, particularly to spike in a long term, but that do not exactly correlate with the neutralizing antibody. So what is your opinion on the implication of these high binders but not neutralizer, particularly not neutralizing the ACE2 interactions?
Yeah, I mean, I think, it's unfortunately our study was very observational, so it's very hard for me to say, again, chicken or the egg, are we seeing very high levels of these antibodies because these patients are not clearing their infections, and so then they have high levels of antigen, and that's stimulating greater and greater amounts of antibody responses, but not necessarily of higher potency. Perhaps the people who have high potency responses, they clear their infection faster, and so then the total amount of antibody ends up fading sooner. So it's difficult for me to say why it is, or whether it's simply a biomarker of disease severity. But I'm not sure I really answered your question, so I'm sorry if I didn't. I'm not
One
here.
last question. Yes, I'm muted it. One last question from I think, if I may, from the audience. I believe there was a question about this in the chat box from Supriya. The question was about this T-cell exhaustion. When we are actually really saying, what we are saying here is that multiple booster doses would probably allow you to better control Omicron or such variants. Is there any possibility of T-cell exhaustions? And if I may add to that, is there any possibility of sort of generating vulnerability to other pathogens in the process of boosting up immune humoral compartment towards a given pathogen?
I don't know. I wouldn't imagine that. I mean, this is not the context of an infection, so I would imagine that just experiencing the antigen 3 times over the course of many months, I wouldn't necessarily anticipate that you would somehow generate T-cell exhaustion from that process. So and I think that no one has seen that being boosted, increases your susceptibility to other pathogens, to my knowledge. So, the boosting seems very safe. So my own opinion would be that probably it's not an issue with the vaccines.
So with that, I believe we can possibly conclude now and really thank Professor Valazis for this wonderful presentation and these elaborate discussions. I mean, we really learn from these sort of sessions every day. Thank you very much. And we will probably have our next speaker of the session. He has not joined. Maybe we will probably take a small 1.5 minutes.
Alex, if you have a cell phone connection for Alex Segal, do you mind reaching out to him? He's actually in Seattle right now. Yeah. If you don't mind calling him, wake him up. It's 6 o'clock. Yeah.
We unfortunately, he was more careful than this. He did not share his number. So
no, Alex Balaz will have it. He's
okay.
trying to.
So yes.
I just mentioned in the chat that Galit will probably cover non-neutralizing antibodies and what they.
Yes, I noticed that. Yes, that would be fantastic to look forward to tomorrow, I believe.
Maybe we can have a.
Yeah, you can. Pushkar, you can take.
No. I mean, because we have to get some talk going. I mean, although
we can chat about things now if you want. It's
although it may be slightly going overboard, requesting you to do what you do in front of your students, which I have done seen innumerable number of times on your YouTube videos, which is the rap and the rhyme of immunology. I know you need to be in the right mood.
I can do anything, anytime. It's up to you.
So please, I mean, we're all ears and eyes, sir.
Let me do something which relates to popular culture then. I can always do rhyme as well, but I'll just do one more general. While Alex tries to contact Alex, we'll see how that goes. Okay, so this is the ode to Ben and Jen, also called Quietly Into the Night. It requires you to know that it's about Jennifer Lopez and Ben is Ben Affleck. But when this was written, Ben Affleck and Jennifer Lopez had broken up. I was informed by my students that they've come back together now.
Yeah,
but I still go with this. I think historically, this will be proven right. Okay. My noble cousin expresses CD4. He is a veritable cytokine store. But a DC's come hither and he's ready to go. His CD40 ligand, he just loves to show. My cousin's promiscuous, hits on passers-by, kisses the wrong B cells, then tells them to die. The right BCR can give fast the slip, shuts off caspase 8 by turning on flip. I grew up a cell of the CD8 kind. I was naive and aimless, bored out of my mind. I wandered like a cloud from node to node. CCL21 determined my abode. Along came a virus, infected the liver. Hepatitis B can sure make you shiver. My DC friends each picked up a sliver. And in the lymph node, I started to quiver. Is this the feeling that Ben had for Jen? Or when Romeo saw Juliet again and again? Peptide in class 1 and then some B7. I leap in delight. I'm in co-stimulatory heaven. I'm no longer a boring CD8 cell. I'm a killer now, baby. A real CTL. No fleeting feelings like Jen had for Ben. I proliferate like crazy again and again. The liver beckons to me and my brothers as well. We each form a synapse with an infected cell. With perforins and granzymes, the little clefts we fill. Oh, what passion to lock lips, but kill. Passion we have now, but once we did grieve for siblings lost, forced to take their leave. They perished At the double positive stage, cell structures, they did strongly engage. They made some BIM and then felt weak. Their mitochondria started to leak. Cytochrome C bound to APAF1. Caspase 9 began to hum. We wept, but they chose not to fight. They vanished quietly into the night. With that, I'm back to science for you guys.
that some memory cells reignited the thing between Ben and Joe.
Yeah, Ben and Jen should have had names beginning with B and T. We do
not have any reply from Alex Segal. That's what.
I forwarded the email to him, and Alex Balaz probably has a cell phone, so maybe
he has tried in the cell phone, but no response. WhatsApp, of course, but
yeah, he says. Unfortunately, I've not gotten a response.
Okay, so you may want to just have a little discussion and close for the day as a possibility.
Yeah, I think. Yeah, I mean, I think this is probably fine. We can there has been quite a bit of conversations on a variety of different topics. And there could be a few more questions. I mean, we had a very good, very thorough 3 talks today. So, and we were unable to accommodate many questions. Some of these we can possibly do. I request the other individuals, including students, please, if you have a question or two, please go ahead and ask them.
Yeah, I think if there are any kinds of questions, happy to answer.
Yes. I think, yes. I think this is a good opportunity. It's not going to happen tomorrow or day after, maybe. Aja, just one more minute, 15 seconds. I need 15 seconds disruptions in the proceeding for some technical issue. Just 15 seconds. So you never know, Alex Segal. Yeah, so hopefully you can hear me, and others who wants to talk, they can unmute themselves. Now we do not have to do it from our end. That was the whole technical thing. But they initially told me that if I do that, Alex Segal will come back. But anyway, so now Dr. Gopalan will.
Can I ask a question?
Yes, Nimesh, you will go fast. Dr. Nimesh Gupta from National Institute of Immunology. You will go fast, have a conversation, and then Dr. Gopalan again. So, Nimesh, go ahead, please.
Yeah, that's a question to Dr. Pillai. Dr. Pillai, we have been seeing that there is a very persistent germinal centers exist in case of infection. Let's say it seems for 3 to 4 months, and from where we think this antigen is lingering around, because definitely it has to stay for long term. The virus is not staying there for the long term for 3 to 4 months. So what is your viewpoint on that? From where it is hanging and from where actually it is coming?
Okay, so I have 2 answers or 3 answers to that. One is for infection and the other is for vaccination. So in the case of infection, there is a paper out by Dan Chertow. I don't think it's in press yet. Who has done autopsies on people who accidentally died after COVID after many months. And he has found virus hanging around for quite a long time, not usually in the lung. So there's a paper from Michel Nussensweig that was published showing that in gut biopsies, up to 6 months after COVID, you still had virus hanging around. So the first thing is that this virus is persistent. Long COVID it could be autoimmune, but it could also be due to persistent virus, which has been found in the brain, in the heart, in the gut. So one simple possibility is that. The second thing is that immune complexes are known to hang around on FDCs for extended periods of time, even in mice. So the half-life of an immune complex bound to an FDC, to an Fc receptor or complement receptor in the germinal center, is quite long. So antigen, even if virus did not persist, and virus is around, it disappears. You cannot find it in the nose after some time, right? But it doesn't mean it's not hanging around somewhere else. So persistent virus is possible. Immune complexes, even if virus doesn't persist, is possible. And the third is a very unusual thing. So I quoted in my talk a paper from Scott Boyd. It's a Rolgen et al. In Cell. Came out 2 or 3 weeks ago. I know that worked rather well. So in that work, there's something unusual that they find, which we shouldn't believe yet. I mean, they don't believe it themselves. They have the data. Well, the data is very good, but you have to still wonder about it. And that is after mRNA vaccination, we not only found protein persisting for many, many months, up to 6 months. So they found antigen only in the journal center. So that could have been FDC, right? But they also found mRNA persisting in the middle of the German center for a very long time. So now the question was, is there some way in which mRNA is being held by some cell, FDC or something else, and is still making protein? Or is it always immune complex? Have we mistakenly assumed that everything we saw in the past was immune complex? Now, I think you can go read that paper. It's in Cell. I think you'll notice that they are careful to say that they don't know how this is working out. There have been very good tools developed to look at mRNA vaccines. There's a monkey paper published from 2 or 3 years ago, which they have now quoted in this version of their paper. And in that paper, they had looked both at the RNA and the protein from an mRNA vaccine, but they didn't look at extended periods of time. So those studies will be repeated by other people. We will get a feel for it. So our understanding of how long antigen persists is not been firm. COVID has started to make us rethink these issues. So one could be persistent virus, two could be persistent protein, the third could be persistent mRNA and protein, and also immune complexes.
Yeah.
Gopalan, yes, please.
Yeah. Thanks, Somen. Thanks, Dr. Pillai. Thanks.
Yes, Dr. Gopalan from National Institute of Immunology. Yeah, please go ahead.
See on screen there. Yes, Professor Pillai, very nice listening to you online.
My pleasure.
Yeah, I was wondering with your new population of CD27 low, CD21 high, and also I'm aware of your earlier work on GVAS, on sialyl transferase and other things. So there is also this, one wonders what kind of antibodies are being churned out by these cells, which could potentially be related to their single cell transcriptomics, or what sort of glycosylation machinery produce a lot of afucosylated antibodies. So do you probably already have the CD27 low cells profile, genomic expression?
We have profiled those cells quite extensively. I mean, I showed you some transcriptomic data on them, but we had been looking at them for a while for another reason, and we now realize that this all adds up together. However, the issue of glycosylation of the antibodies, I have definitely not looked at, and Galit may talk about it on Friday. Okay. She is interested in the question of the different glycosylation of antibodies, especially the question of afucosylation and so on. I mean, we're thinking in the field which is a little rough and not well established. I mean, she may push that line. I don't know. I don't think it's that well established. Is that maybe in some types of infections, you cause the generation of antibodies that would help you. NK cells would be helped by certain antibodies, so maybe you have glycosylation changing. I think that's an interesting area without very firm conclusions that can be made yet, but it's interesting.
Sure. Yep. Thank you very much. Thank you.
I think I believe it was nice to really get this extra space to have this extended discussions, and we probably will close the session for now. We will start tomorrow Indian time 5:30 for all others, and the link that we have shared with you is sufficient for you to have multiple entries for all 3 occasions, all 3 days. And I sincerely thank Professor Pillai and other speakers. I mean, we understand that the time zone and then the difficulty, that the awkward time of the day that they have to wake up and give this talk. No word is sufficient. And we learned a lot from today's session, and also other people who are with us. And I will leave Dr. Pushkar Sharma for a few words before we conclude the session today.
I think famous last words have been spoken, and I don't want to, put any other thoughts together or forward. And we look forward to your participation tomorrow. And Professor Pillai, it was great. And I mean, I just don't want to be repetitive and boring, but the fact remains is that I'm greatly indebted to you for putting this together, and your passion and the interest is evident, and and it's rubbing on to us. Thanks very much. And we'll see you tomorrow. Hopefully, Professor Pillai, you'll be there tomorrow as well.
I'd like to thank Alex for at least one Alex for participating today. Thank you.
Yeah, thank you so much. Sir, I think you spoke too soon when you said we have 4 Alex.
So we're down to 3 now.
But we can be 3 is a good number, sir. Thank you so much. Thanks, everyone. Have a good night or a good day wherever you are. Take care. Cheers. Bye. Yeah.